Okay, so hopefully that gave you a little bit of a look at some of the things we do to physically see what it is we actually look at when we say we do stuff. So now the field is yours, and you can ask any question. So we may as well make use of the microphone so that people can hear the questions that get asked, and then I'll do my best to answer them. So.
What will it take for, because I, and that's like a mother feeling, like, and I think I told you yesterday, there's this like feeling that the gut and the microbiome is sort of working its way. I know it works with everyone, but in this case with the disease, I think it's like taking a toll on it. So what would it take to take investigation there, like?
Yeah. So that's a great question. We spent all our time originally just looking at the brain. Then, because I listened to lots of families telling me their kids had problems pooping, I started investigating the bowel, and I found that there were a series of problems in the bowel that are a bit similar to what happens in the brain and a bit different. So we have neurons, nerve cells in the brain are having a problem and they're dying. The same is happening in the bowel. Those brown cells we saw down the microscope was the brain's immune system getting activated and responding. Something similar happens in the bowel, but the actual types of cells are a little bit different, yeah? And the immune system in the brain is kind of sheltered. It's behind a blood-brain barrier. In the bowel, it's wide open, and everything that's in the blood or in the bowel will influence it. And one of the big things that is very hot topic of debate is the gut microbiome. So we have billions of bacteria in our bowel, and there's evidence from lots of diseases that the composition of those bacteria can have an influence on the brain and can alter health. And for example, in Parkinson's disease, there's now evidence to suggest it actually starts in the bowel and then spreads to the brain. And there's similar evidence for Alzheimer's that something is going on, that the bowel is a big influence upon the brain, and vice versa. Yeah. So the question about how do we investigate whether the microbiome has anything to do with it, that's a question I always get asked when I start talking about bowels, because everybody's excited about the microbiome and we can measure it very well now. So we've started doing that, and we've done it for CLN2 mice so far, because we were focusing on one type and CLN2 mice progress very quickly, so we can do those experiments quicker in a CLN2 mouse than we can, for example, a CLN3 mouse or even a CLN1 mouse. So we've done those experiments. We know that the composition of bacteria, the microbiome in a CLN2 mouse, is different to a regular mouse. And now that becomes a question of chicken and the egg. Is the changes in those bacteria part of the disease, which is the question you're asking, or do they change because of the disease? So we've done an experiment to actually do that, and the way you do that is you wipe out the bacteria by giving our mice a load of antibiotics. It wipes it out, and then you see what happens to disease both in their bowel and in their brain. And you would expect that if the bacteria are up to something, they're causing part of the disease, it might change it. Either one way, or it might get worse, it might be less severe. And the answer is, when we did that, actually nothing changed in the bowel and nothing changed in the brain. And whilst that's only one experiment and a very simple experiment, it tells us that probably the bacteria in the bowel are not driving the disease, but they're changed because of the disease in the bowel, probably. But they're not causing the problems in the bowel tissue itself. They are changed probably because the bowel's not working properly. And that's important to find out, because we need to know if it's doing something important or not if we want to try and manipulate it, because you can manipulate the microbiome easily by drinking, yogurt drinks. Excuse me. or probiotics or something like that. So all our work is suggesting that, yes, the microbiome is changed, but it's not driving the disease. Doesn't mean it's not important, but it doesn't mean it's the root cause of it.
Okay. So like to kind of piggyback on that, is there a connection between like absorption with our ceiling kids? That's why this sort of bacteria thrives with their kids and maybe others don't. Is there an absorption of certain types of things?
So that's, I mean, it's like you're reading my mind. Yeah. So when we found there was a problem in the bowel, we first wanted to say, was there a problem in the bowel of our mice, and was it anything like we see in people? And that's why we did those tests of seeing how fast things move through the bowel, and we saw it went slowly. And that's when we started looking at the nerve cells in the bowel, because they control that whole process where food moves along your bowel, something called peristalsis, has to be a coordinated series of squeezing and relaxing. It squeezes here and relaxes the next bit, and then that moves along. That's what's controlled by all those nerve cells in the bowel. So we thought we were clever when we found that out, and it was exciting. But the more we look at the bowel, the more we see is wrong. So we've just got a big grant, which is all about the bowel and how the bowel relates to the brain. And the things we're doing in that, to get a big grant like that, you have to do some early experiments, preliminary experiments. And we found that the surface of the bowel, which is where the absorption happens, those villi, those fingery looking things, are actually much shorter. And those cells that line the villi come from a deep down structure called the crypt, and that's also different. So we're guessing that if the cells that do the absorption, the area for that is much smaller, probably absorption will also be changed. And when you have absorption problems is when you have various stool problems. So we don't know. At this point, we don't know. But what we're finding is the more we look at anything, the more different it is. So within the bowel, we started off only looking at the nerve cells. Then we look at the nerve cells and the immune cells. Now we're looking at the cells that are doing the absorption. And we're also looking at the muscle in the bowel. That also has a problem. And that's what we're going to do in this grant. And I can't tell you the answers. But that's what we will be doing, is looking at all the cells. So Katie yesterday talked very nicely about looking inside cells to see which genes are switched on and switched off. And she's doing that with the brain's immune system currently. But we will be doing that with all the cell types in the bowel to find out which ones are affected. And then, practically, what problem does it cause? Yeah, so we have.
I don't know if this all ties in together, but how and would that be related with like the dysautonomia or autonomic dysfunction that we see, like when my son had the GI tube placed and his CRP was through the roof? There was no infection anywhere. And they were baffled, and I was like, this is Batten disease. So that peripheral nerve system that's also playing into, these random fevers that have no cause, the vomiting that has no cause. So is that perhaps not necessarily all those signals are coming from the brain? They're also,
So it's an excellent question. The whole idea of the autonomic nervous system being affected, in particular in CLN3, is a very new topic, one that's emerging. And there's a doctor in Denmark called John Ostergaard who's been working on the autonomic nervous system and imbalances between the two parts of the autonomic nervous system. So there's a sympathetic and a parasympathetic, and how they are in imbalance and how that causes fear and anxiety and various other panic cat-like behaviors. So he also was the person who first described heart problems in CLN3 disease and has been looking at that. And we're also looking at it. But the whole idea of the autonomic nerve and its influence upon the bowel, the vagus nerve, is something that we're also very interested in looking at and finding out what's happening. And most of this is now just looking at the disease with a very different pair of eyes. We spent all our time just looking at the brain and the spinal cord. Now we're looking at all these other things which common sense tells us they're probably all affected, but we don't know how. And that's what we've kind of opened a new can of worms. And the more we look, it's like like I said, we started in the bowel with one type of cell that we were interested in, and now we're looking at all of them. So there's lots of things. But yes, the autonomic nervous system is important for how the heart works. So yes, it's going to be. Mm-hmm. Yes, yes. So all of these things will need looking at, but they're going to need looking at systematically, carefully, and to be able to do that, we need money, and that's why we have to write the grants to get the money to do that. But you know, we're lucky that we just discovered on Friday that our big bowel grant was finally awarded. Yes, it arrived. It was held up in the mail for some time. We got a really good score on it in February, and it took till July for the. I think there might be a lot of strange communication going on. Yes. So, happily, we are now in a position we can start asking these questions.
So I know that a lot of the focus of research is finding something that works, a treatment that works, but then you have to deliver it. And so I have to like, one of my greatest worries, right, is that like an effective gene therapy will be developed, but if it's delivered on the AAV9, that my kids will have had an infection somewhere and they have an immunity to AAV9. So that's a long introduction to my question, which is what kind of work is being done to look at different delivery methods and like what's effective for those different parts of the body? Is there something other than AAV9, which is not a perfect delivery system?
Well, AAV9, just so gene therapy, to back all the way up, so gene therapy is taking a virus and using it as a delivery system to express what's missing, a functional copy of the gene. And there are lots and lots of different gene therapy viruses, some of which fall in one category, some fall in another. The ones that are being used most commonly for gene therapy therapeutically are AAVs, which stands for adeno-associated virus, and they come in different flavors. They have different serotypes, which are numbered 1 through at least up to 9, and AAV9 is the one that's been used most. Now, in itself, you have to then get that to where it's needed, because that has to go inside the cells to be able to express the gene that's not working, to be able to make the protein. And there's slightly different requirements for that, depending on whether you have a protein that's released, like CLN1 and CLN2, or whether it's CLN3, because you want to try and get it into more cells then. And the properties of those viruses are really important. Also, where you deliver them is really important. Now, we can test out ideas of where to deliver them, and we test them out in our mouse models or in our sheep models. So, you know, in our grants we say, do I put it in here or do I put it in there? You know, and, you know, in our sheep it's like, do I do it by lumbar puncture or do I put it into the ventricle at the top of the brain up here? And see which is the better way to do it. And that's the sort of thing we do. That will tell us where is better to put it in, but the actual properties of the virus itself become really important. And that's where we learned something from people who are, their living is changing viruses to make them have better properties, or properties that will be more useful for doing therapy. So they might spread further if you put them in. They might express the gene that's missing really well when you give less of them. Because one of the issues is the more of a virus you give, the more of something will be made, but the probability of there being a side effect of that also goes up. So if you could achieve the same result by giving less virus, just a better virus, that might do better. And that's the approach that Lattice Bio are basically doing. They've engineered the outside part of the virus, which is something called the capsid, so that it interacts with other cells in a more efficient fashion, or it will spread further. So it's going to be a combination of basic nuts and bolts knowledge of the disease and some common sense as to physically where you put it, but you're still going to need to develop probably better tools than the ones we currently have. And it's encouraging that those are happening. You know, and Lattice Bio was founded by the person we collaborate with to do our gene therapy. And why did we pick that person? Because they're very smart and they're very good, and we've known them for a long time and we trust them. You know, so we work together, and we've known each other for many years, and we choose to work together because we can do some things that they can't do, and they can do many things that we can't. But by tag teaming together, we're better able to come up with a better tool that we're able to do it. And we think that testing it in a large animal model is important for knowing how it moves around in a larger brain. And that's what they've done. They've taken their viruses and they've engineered them in a particular way and tested them in a non-human primate, so effectively a monkey of some sort, which has a much bigger brain than a mouse, that is more like a human brain. Even though it's called a non-human primate, it's a complicated brain. And they found they published an exciting paper on this recently, which says this is what these viruses do. And that would give us some hope that they might work well and work well at lower doses so that there would be less side effects. Now, will they? We don't know yet, because some of those experiments have been done in animals and they look good, but I'm not going to talk about you talked about it. It's, you know, working towards actually doing that in people is the next step. But the question you said about if you already had an AAV, that's a problem. So about 30% of the population already have antibodies. Well, you talked about antibodies as tools to find things, but the immune system's defense. About 30% of us have antibodies against AAV9, for example, because we had some infection at some point. Yeah? And an AAV is an adeno-associated virus. It's associated with an adenovirus, and that's what's causing things like colds. So if we've had a particular type of cold, a particular type of fever, we may have an antibody that's already present. So people are also, you know, and that potentially means that if you were to give a gene therapy virus, the immune system would recognize it and it wouldn't then work as well, or there'd be a big immune response. So people are also working on, what do I do if that happens? Yeah. Now, the other big thing with gene therapy and the immune system is it's supposed to be a one and done. Yeah. We hope that you will put gene therapy in and that virus will keep making what's missing forever. Will it? We don't know. Will the expression stay at the same level? It looks good over a period of time it's been tested over, but who knows how long it will be in 10 years' time, 20 years' time. So what you might one day have to do is to give a second dose. Now, if you were to give a second dose, you know that that person would already have many antibodies against that virus because we've given a lot of virus. So they will have made antibodies against it. So if you try and dose a second time, that could be a problem. And that's why people are working on ways to manipulate or suppress the immune system to make things like redosing possible. or the dosing of children who happen to have naturally occurring antibodies possible. Because, you could find yourself in a situation where this therapy's been developed, you want to be in the trial, and you discover you've got the antibodies. Yeah? So there has to be some way to try and intervene with that to make that treatment possible, or retreatment possible. So people are working on that. Just like I'm not the person to mess around with the capsid, the outside part of the virus, that's not in my skill set. Other people who have those skills are doing it. Other people are working on the whole immune response side of it.
So with the gene therapy and CLN6, Kristen had told us, that when you give it, only a certain percentage of cells are affected. And I want to say, I may be making this number up, but 30, for it to see a difference, it has to meet a certain threshold. I think she said 30%. I don't know, but is that true? Is there a threshold that has? And does it over time, will that make a difference in the long run in terms of stabilizing?
So that's a good question. So we're using a virus as a delivery tool for something that's missing, and that something is missing from every single cell in the body. When we give a virus, a gene therapy virus, it's going to get into some cells. If it was the perfect virus, it would get into every single cell and would make what is missing in every single cell. The current viruses that are being used are good, but they're probably not the perfect virus. That's why we have people who are devoting their careers to improving them and founding companies to improve them. So in what was used, that was a regular AAV9 virus. There was nothing especially fancy about it from what I know. It would have got into a proportion of the cells. Then the next question is that a problem that it's only in some of the cells or not? It could be, but it could not be. So if it was CLN1 or CLN2 disease, cells that got the virus could make it and release it, and it would be taken up by their neighbors, and it would fix the problem. That's something called cross-correction. Now, because CLN6 isn't a released enzyme, it's stuck in a compartment inside the cell, it's not going to be released. So what you really want is to get more cells with more virus inside them to make it to have a better effect. Now, we don't know what the threshold is that you need to reach to get the best treatment effect, but we know that it's better than not having any. Yeah? So with current tools, I mean, it's interesting, all the stuff we've been doing, we do gene therapy in our mice for CLN1, 2, and 3. And some of the results we get for CLN3 are surprisingly encouraging. Even though the virus is not getting inside all the cells, it might be getting inside the right cells or some of the important cells. So, and that comes from understanding why certain cell types are more or less important than others, and whether you could make a virus that would go specifically in those cells. So all of that is also happening and working along, but in terms of the gene therapy for CLN6, when it would get restarted, you'd have to look at playing with the dosing. Yeah? You'd have to change the dosing to be able to get more virus into more cells. Yeah? And that's what they learned from the first go at doing it, was that you may have to change it. Yeah.
So it's going back to the bowel, and I guess there must be a difference between doing the studies in CLN3, CLN2, and how it would work for other CLNs, like for example CLN6, right? Or could you assume because it's happening this way in 2 and 3, then for the rest it must be the same? Or
So the answer is it will be the same but different. because it's a different gene. Yeah, but so for example, we're now looking at lots of diseases that affect the brain, similar diseases, and saying, does it also affect the bowel? And the answer is yes and no. So we're part of a big group at WashU, which they built us together all to be in the same place. That's why I was dragged away from UCLA to go to St. Louis, was to build a group of people, all who work on lysosomal storage disorders. So there's about 70 different diseases in which the lysosome, the cell's waste disposal unit, goes wrong. And, you know, there's 13 or 14 of those that are different forms of Batten disease, but there's lots of others. So we've talked to some friends who work on a similar but different set of diseases called mucopolysaccharidosis, or MPS, where many of the families face very similar practical problems, and we've been starting to look at MPS mice that also have some brain problems, and do they also have bowel problems? And at the moment, it looks like the amount of brain problem is sort of proportional to the amount of bowel problem. So the MPS mice we looked at had far less degeneration in their brains, but they also had far less problems in their bowels. So we can't assume they're all going to be the same. We will look at CLN6 mice, but you know, we have to get money to look at CLN6 mice. So we will be doing that work for sure. I would, if I was a betting person, which I'm not stupid enough to bet, I would guess that they would have that problem because they have enough problems in their brains. And if it's as simple as my simple thinking, which is if there are brain cell problems, there will be bowel nerve cell problems, then I would say that it's very likely that there will be a bowel problem. But I can't tell you for sure until I've done those experiments, but rest assured I will be.
Something else. And I don't know if it's like, if the question is for here, but sometimes I think like all the things that happen around us or to us with our kids, and I can help but think that before I had my son that has CLN6, I had another pregnancy that I had to terminate because the baby had a brain malformation that was not life compatible. So I'm always wondering, and I sort of like have heard from other families before having their kids, or in between kids, they have pregnancies. So could there be like any correlation, like, or is it just like maybe coincidence, like, yeah, I lost my first baby and the brain was the same thing that was damaged?
So the answer of that, I mean, it's a great question, but the answer is we can't tell, and you couldn't tell without actually studying. You know, there are lots of reasons why kids have issues that means they don't come to term. And it could be something related, but it could equally be something entirely different. And you don't know many pregnancies that don't go through naturally, never mind needing to terminate them. So it's almost impossible to answer that. Now, there has been thoughts that mistakes in these genes are clearly a problem. You know, I mean, so for example, we still don't know what CLN3 does. But we do know that whatever it does is really important, because when it's not there, you have a very serious disease. Yeah? But that's not having it there at all. There are some people who say, if you completely took away CLN3 totally, not even making a form that was much shorter, would that be compatible with life or not? And some experiments have been done in cells in a dish, which might suggest that there would be severe issues. But whether that's true in life and whether it's true in people is not clear. So unless you actually were to directly study that, you would not be able to answer the question. But it does make you think. Yeah?
We have so many questions.
I know. I've been this way since I was a kid.
Anyone else who wants to stop them from asking all the questions?
Yes, please.
Go ahead.
No, you go ahead.
Okay. Do y'all find that there is, so my daughter had, early on, they were found, it was an incidental finding that she had, a cardiac defect. It was the vasculature, nothing that was like, there's a lot of really terrible ones, and it wasn't those. Do y'all find like other genetic or like other anomalies with these children, or is that something that's just a coincidence?
So that falls into almost exactly the same category as this question, unless you actually investigated for other mutations. So all of us have 20,000 genes or thereabouts. Of those genes, a couple of hundred of them maybe have a mutation in them. That may be a mutation that isn't causing a particular problem, and it's only when you have a kid with someone who happens to have a similar mistake with one of these diseases that you end up with the problem. So the answer is, unless you specifically hunt through all your genes and find out which mutations you've got, you'll never know whether it was a coincidence or whether it's a specific thing. Yeah? And I have absolutely no problem if you ask all the questions, but
I had one, but you answered it in the course of earlier.
Okay.
Well,
I guess I got one, but I don't know if it's a silly question or not.
there is no such thing
Because I'm
as
not much in the
there is
understanding biology and stuff.
question other than will the Dodgers win the World Series?
They will probably, likely. The Detroit's doing good too. So if we got DNA is everywhere in our body, right? And if these diseases see on all of them, you would be able to see it in every part of the body, basically, because the DNA is messed up. So.
But you don't see the effects of disease everywhere.
Yeah, I guess that's why. Okay, that was kind of my question. Like, the DNA is messed up everywhere.
So there is no such thing as a stupid question. That's just a question we don't know the answer to. Yeah, and that's a tricky question to answer. Now, yes, all cells in the body of a child with Batten will have the same DNA problem. Yeah, but will that actually cause a real problem to those cells or not is harder to tell, and it's partly down to the fact that cells have a naturally different lifespan anyway. So, you know, in our homes, the dust is mostly made up of dead skin cells. Most of our poop, or at least a large proportion of it, is made of the cells that were lining the bowel and fall off and get replaced by new ones. So if you have a cell that doesn't last for very long and it has a biological problem, you don't get to see it because that cell dies and gets replaced. Yes, the ones in the brain last for as long as we do. So there are areas in the brain, some very small areas, where something which is called very fancy neurogenesis, which just makes making new neurons, happens, but it's not very much. So once we've gone beyond sort of teenage years, late teenage years, most of our brains have got all the cells we're ever going to have. And you have to make do with them over a long period of time, and they have to last all that time. They don't divide and new ones come along to replace them, but as we get older, they will get more dysfunctional over time. The other thing about nerve cells is, compared to, say, a skin cell or a cell that lines the bowel, they're a lot more complicated. You can think of, for example, a liver cell as being a little round cell. A nerve cell can have a projection which goes from here all the way down my spinal cord and then out down my leg. So it can be, you know, over a yard long. And that's a complicated cell. It's got to receive information from lots of places, so it has long, thin processes to do that. It's got to send messages all the way down that really long distance, and to be able to communicate, to make me move my foot around, it has to fire a transmitter, a chemical messenger that says, "Oi, you, move." Yeah? And that has to be, that's made up here, and it's got to get down there. That's remarkable that it ever makes it there. And that's basically what you've got, is cells that are really complicated that have to live as long as we do. So it's no surprise they might go wrong. And one of the reasons why they're going wrong is that the lysosome, which everybody thinks of as being the waste disposal unit or recycling center, is actually also really important for how things move around inside a cell. So when that goes wrong, so nothing that comes into a cell or goes out of a cell can do so without going through the lysosomal endosomal system, which is basically what's going wrong. So if that system, which is important for moving things around, is screwed up, how things move around inside a cell is also very likely to be screwed up. And if you've got to do that over a very long distance with exquisitely perfect timing, then it's not a surprise that it would go wrong. It's probably more of a surprise that it doesn't go wrong more often. Yeah?
And the reason why,
Mhm.
you start developing symptoms later is because it was going right.
Yeah.
And then it stopped.
Yes. So that's, I mean, that's a really good question which comes out of that, is how come kids who have Batten don't have the effects of that from the moment they're born, and you can't see it? It's because the system is kind of good at coping, and it can cope for so long, and then it gets less good at coping, and then you start to see. And the reason it's different for different forms is because it's a different gene and a slightly different problem. And that's why we have, you know, an infantile, CLN1, use the old terms, infantile, late infantile, juvenile, adult. They start at different ages because it's a different problem. But to go back to your original question, like, do we see effects of disease in the rest of the body? Well, now we're actually looking at all of those. You know, we've spent about 20 years looking at what's going on in the brain, and it's like, why didn't we look at the bowel earlier? And it's kind of embarrassing because to get the brains out of our mice, we go through their abdomen to their heart, stick a pipe in the heart, rinse the blood out, and send a fixative round to preserve it. But to do that, we've gone past the bowel, and I could show you pictures you don't want to look at where the bowel is full of fecal material, and it was there. It was staring us in the face, but we never looked at it because we must look at the brain, have to look at the brain. We'd ignored everything else. But if you now look at those mice, you can look at them and go, yeah, that one's affected. No, that one's not. Oh, the treatment's worked in that one. You can tell just by looking at how much poop they've got. You know, and should we have noticed that 20 odd years ago? Probably, but we weren't looking for it because we just ignored it and went straight to the brain.
It's not really a question, just an observation. I mean, the first thing that landed us in ER was severe constipation after diagnosis. It wasn't the seizures or anything else. It was like the pain of not being able, and he was just, and repetitive, you know, and we were constantly, thankfully we had a pediatrician who referred to himself as the poop guy because he has a brother with autism. So that also sometimes has an effect on motility. And so he was really good at that. And instead of all of these different powders and Restorelax and things like that, we went with the magnesium citrate liquid, and that made a huge difference. That was so much better,
And this is one of the frustrations for kind of us when we're trying to work on this completely new area. I say it's like the Wild West down there, you know. Once upon a time I was a neck-up scientist. Now I'm also a bowel scientist. And that's because biology's cleverer than me. Well, actually, Batten parents are cleverer than me and told me I had to look there. But you can get the SIBs on your side in seconds. All you have to say is poop, and they're like, "Whoa, where?" You know, and that's you know, immediately you're like the coolest guy on the planet, which for me is, you know, something of a miracle. It's you know, but these things are all there. If you ask anybody at this conference about pooping, everybody will say yes, there are problems. Now, is there anything published on that in people? No. So when I had to talk to my first bowel dude to try and get him to help me, I had to say, look, I work on this kid's disease that attacks the brain, and all the kids can't poop. And he's like, where's the evidence? Where's a paper that says the kids can't poop? And there isn't one. There isn't one at all. You can look in a review that talks about the whole disease, and there might be one line that says constipation or bowel problems. And that's why we're now starting, and hopefully you will hear about in some point, once we get to that point, we want to actually survey families to tell us about all these problems so that we can actually build some evidence that this is really happening. And what we want to do, and this is where we have to pick our friends very carefully. Unfortunately, I picked the best guy, who's somebody called Bob, who's at CHOP in Philadelphia. He's a really cool guy. He works with kids who have bowel dysmotility. He cares deeply about them. He's a really good person. And he has worked in a variety of other diseases where they've done a similar thing. They've done questionnaires of families to look at all the different GI problems that you might have. And, you know, GI problems come in lots of sorts. There's constipation, there's diarrhea, there's reflux, there's vomiting. There's all these different things, many of which are happening in all the kids, but we don't know about them or how they might be related to one another. So we're busy building information about that. And that's what we want to do. So I'm talking to clinicians who work with kids with these diseases, trying to get them to look through their records of what notes do they have about an ER visit because of a constipation problem, or when did they prescribe some particular drug that was meant for a bowel intervention. And by doing that, we hope to actually build a proper story about the disease in people. Now, we do know, A, that it's happening in people, because you told us, and I'd have been really stupid if I didn't listen. Yeah. We also have all these sort of anecdotal reports, but we've also actually looked at the bowel of children. Yeah, so we have looked at the bowel of a child with CLN1 disease and of a child with CLN3 disease, and this was material that was taken after that child passed away, and we had permission to do that. And we talked about tissue donation yesterday, and most of that was a focus on the brain. We're also looking at bowel now and requesting, if people make the decision to donate, that we will also want to look at bowel, because we want to be able to look and see that this is really happening. And all our work says it's happening in people, and remarkably, how it looks in people at a cell level is remarkably similar to what's happening in the mouse. There's a very unusual thing that happens with the patterns of cells dying in the bowel, in that you have little groups of bowel cells. They're called myenteric plexus cells. They're in little clusters called ganglia, and you can see some of them look perfectly fine, and others look totally wasted, totally devastated. And in the areas where they're devastated, that's where the bowel's immune system is doing something. And it's exactly the same in the human samples as it is in the mouse samples. We'll get to the bladder. So the bladder is actually a big problem. So I mentioned the MPSs, the mucopolysaccharidoses. Bladder issues are a really big problem there. And the mice often die because they have a bladder that's massively full of urine that doesn't get released. So, yes, so all these things will need looking at. I've got to get busy writing more grants. About hiccups. Nobody has told me about hiccups, but now's your chance.
No, I guess it's because we have the big population of CLN6 in Costa Rica. And there is this one mom that her girl passed away like 20 years ago. She wrote this book, and in the book, or a little pamphlet, and she wrote some of like the symptoms or signs that they saw. And for some reason, she wrote down hiccups. And I see hiccups in my son since he was born. Like, and when I read it, I asked her, like, really? Because I thought this was just like something. And she said, yes. Like, we've seen here in Costa Rica that many kids with CLN6 will, for some reason, have hiccups. I don't know if it's also GI related.
So hiccups are kind of both a bit of autonomic nervous system and a bit of gastrointestinal thing. It's a bit like I mentioned yesterday, the swallowing things we're doing. The swallowing thing seems to be a combination of all the things we work on. There's some pathology in important places in the brain. That's where the nerves that go to the muscles involved in swallowing come from, or they receive information back from them. We know there's a problem there. We think there's a problem with the nerves themselves and their endings. We think there's problems with the muscles, and there's also the esophagus, which is, going from the back of your mouth down to your stomach. That has muscle layers. They have a problem. The tongue also is atrophied. And there are some nerve cells in the esophagus. They also have problems like we have in the bowel problems. So it's like a combination of all these things. And that's what we're now working on with the grant we got recently. So, but probably hiccups will be autonomic, I'm going to guess.
I missed your question because we got chatting, but it was like they would be repeated, and it would like take a really, really long time to settle down. And we always thought it was like some issue with the diaphragm, that it's just kind of like getting stuck almost. That, yeah.
So we've covered a lot of ground. You probably weren't expecting the brains. If anyone wants to look, so we're kind of at the end of our allotted time. I don't mind carrying on a bit longer, but you may have other places you need to be and other sessions to go to. But if anyone wants to hang around and have a look down a microscope, they're still here, and we can carry on a bit. Otherwise, I'm going to be here till tomorrow morning. If you see me sitting there staring into the distance, I may be worrying by how much the Dodgers have beaten the Giants, which is going to happen this afternoon and again tomorrow. Yes. Yes, this afternoon. It's happening in about half an hour. Okay, thank you everybody for coming, and thank you for questions.